Hello, in this video, we are going to be talking about mitosis. So mitosis is a type of cell division where the cell is going to divide in half, and then we are going to produce identical daughter cells. And here in this picture, I am showing you a picture of the DNA of a specific person. And as you can see, their DNA is organized in chromosomes. We'll talk about that in this video. But what I wanted to emphasize is that our cells have a lot of DNA. And for the cell to then replicate that DNA, so now you have double the DNA, and then organize it so that you have split it perfectly, equally, is a difficult task. So DNA, very large molecule, you have double the DNA and you're trying to evenly split it so that when you divide, both cells have identical DNA. That is very difficult. And so that is why mitosis has so many different steps. And that's what we're going to be talking about. So first and foremost, the DNA has to be packaged. Remember that DNA is this long strand. It is a macromolecule, so it is super humongous. And, just like any kind of string or thread or cord, I'm sure you've experienced that these things tangle very easily. So, things like your headphones or even just like people who have long hair, right? So things that are long and stringy are going to get tangled relatively easily. And so what the cell does to make sure that its DNA is, easy to move around is that it packages them into chromosomes. So during S phase, and so this is from the cell cycle video, during S phase of interphase, the DNA gets replicated. And again, now we have double the DNA. And then later on in the cell cycle, the DNA will get packaged, basically coiled up into chromosomes. So here's your DNA. It's going to get coiled around histones, and then it basically loops around itself a bunch until it produces this structure called a chromosome. And so typically drawn like this. Now, when your chromosomes are replicated, you're going to have two halves, and they're called the sister chromatids. So this right here is one sister chromatid attached in the middle to another sister chromatid, and they are identical to each other. By the way, if we were to draw a chromosome that is unreplicated, this is what it would look like. So you just have kind of a single line. But when chromosomes are replicated, which is after S phase, you're going to have two sister chromatids that are combined in the middle. So it kind of forms an X shape. Now, it gets even more complicated because each person, at least in humans, each person contains two versions of the same chromosome. So, for example, we typically number the chromosomes to make it easier. So I have, chromosome number 1, chromosome number 2, all the way through 22, and then the sex chromosomes. And each person has two versions. So this diagram right here is showing non-replicated chromosomes. So this is one chromosome, this is another, this is one chromosome, and that's another. So they're not attached, okay? So I know a lot of people think that they're attached in the middle, but this right here is what we're looking at here. And each person has two versions. Why? Because we get one from each parent. So I got chromosome number 1 for my mom and another chromosome number 1 for my dad. Chromosome number 2 for my mom and chromosome number 2 for my dad, and etc. So each person has two copies of each chromosome. So that makes it even more complicated because we have so much DNA and so many chromosomes in the cell. And again, in this diagram, we are showing non-replicated chromosomes. Now imagine this diagram, but each chromosome is actually in its X form. So we have so much DNA. And again, that is why in mitosis, the cell really works hard to make sure that it is equally splitting its DNA in half. There is a lot going on, lots of DNA, lots of chromosomes to basically sort out. So yeah, that is the first thing that the cell does, is it organizes its DNA into chromosomes. So let's go through the process of mitosis, starting with part 1. And so this is actually before mitosis. We're going to just recap what's happening in G2. G2 is the last step in interphase. The cell is preparing itself for cell division. It's making itself really big, making sure it has tons of nutrients, double checking that its DNA is replicated. Now we're going to go into prophase, which is essentially the first step of mitosis. And in prophase, the DNA gets condensed into chromosomes. So right here, the DNA is all over the place. And the DNA is going to start coiling up into chromosomes so that it's easier to move around. Another thing that happens is that your centrosomes are going to start producing the spindle fibers, also known as microtubules. And the nucleus starts to also kind of disintegrate because, again, we are trying to sort the DNA, and we're not just going to do this in the nucleus. We're going to open the nucleus up so the DNA is now out and we can organize it. Okay, then we move into prometaphase. In prometaphase, the chromosomes are going to bind to those spindle fibers. So these spindle fibers are being produced by the centrosomes. They're basically really long microtubules. And here you can see that the chromosomes are actually binding to the microtubules, or vice versa. So the microtubules are grabbing onto the chromosomes, and they're going to grab onto the chromosomes in the center. And notice that, especially for this chromosome, this one is attached on one side to one microtubule and then on the other side to another microtubule. So basically, both ends of the chromosome are being attached to microtubules on one side and the other. This one is still in the process of doing that, but eventually they will all be attached to microtubules. Okay, so that was prometaphase. Now we're going to move on to metaphase. In metaphase, all of the chromosomes are going to line up in the middle, and notice that each chromosome is attached on both ends. So there is a spindle fiber here and a spindle fiber here grabbing onto the chromosome. So they're basically sandwiched. And all of the chromosomes line up in the center. We call this the metaphase plate. Again, we are trying to equally divide our DNA. It'll be easier if they're all organized in the middle first before we separate and split it. And that is essentially what happens in anaphase. So in anaphase, the sister chromatids are going to separate. Okay, so remember that a chromosome, when it's replicated, it looks like an X, right? And each half of the X is a sister chromatid. Those are identical to each other. And what ends up happening in anaphase is those microtubules shorten and they pull the sister chromatids apart. And that is what we're seeing here. And so now you just have equal halves being pulled apart. At this stage, those chromosomes are called daughter chromosomes because now they're separated. So they're not called sister chromatids the moment they separate. Okay. And then finally, so we've divided our DNA equally in half, not really in half, but we've split the DNA in half. And so now the cell can reform its nucleus. So it's going to reform one nucleus here and another nucleus there. And then during cytokinesis, the entire cell kind of splits in half. All of the other organelles get split. The cytoplasm gets split. And then the cell membrane splits. So that's that. Notice that, again, in mitosis, the majority of the steps are dealing with splitting the DNA. The DNA is, again, the main focus here because we have so much DNA and we want to make sure that we're equally splitting it. And then everything else happens later. All right, so that's it for this video. The last thing I wanted to mention was that in plants, when we're doing this cytokinesis, the cell essentially pinches itself in half. So there are these proteins that tighten in the middle, and essentially the two cells split through a pinching motion. Whereas for plants, instead of pinching, the plant cell actually just forms a cell wall right in between the two cells. And so there's no pinching involved. They're basically just building the wall in between. All right, that's it for this video. I hope this was enjoyable, and I'll see you in the next video.